When we talk about the process of alpha-ketoglutarate being converted into succinyl CoA in the TCA cycle, we use an enzyme known as alpha-ketoglutarate dehydrogenase complex, which is made up of three subunits, E1, E2, and E3. Now, this mechanism is going to be very similar to the mechanism we've already seen of the pyruvate dehydrogenase complex, which is also a complex with E1, E2, and E3 subunits. So the mechanism is going to be similar to that of pyruvate dehydrogenase, but we're just going to use different molecules in this case. When we talked about pyruvate dehydrogenase, we were using the compound pyruvate, and we were converting pyruvate into acetyl CoA. But when we use alpha-ketoglutarate dehydrogenase, we are going to convert alpha-ketoglutarate into succinyl CoA. So essentially, the mechanism is the same, just the molecules are going to be different. So we begin our journey of this mechanism. To just get a quick overview of what we're going to do, we begin our journey with alpha-ketoglutarate. And the first thing we have to do is we should count how many carbons we're dealing with. So we have carbon 1, 2, 3, 4, 5. So we have 5 carbons. In the process, we can see that we will be losing 1 carbon as carbon dioxide, and we will be adding in coenzyme A into this reaction in order to get succinyl CoA, because we can see over here we're adding this coenzyme A to get this thioester bond in the end. So that's important. What else is important that we should make note of? Well, we're going to be reducing NAD plus into NADH and H plus. And then once again, we have 3 prosthetic groups. So we have FAD, we have lipoate, and we have TPP. Now, these 3 prosthetic groups have specific locations in this complex, and they are covalently linked to the enzyme. So we're going to find in E1, so essentially alpha-ketoglutarate dehydrogenase complex is made up of 3 subunits. So we can call this subunit 1, and in subunit 1, we are going to find TPP. This is E1. In subunit 2, so E2, we'll find lipoate. In subunit 3, we are going to find FAD. Okay, so alpha-ketoglutarate dehydrogenase complex is a large enzyme, and it has 3 main subunits, E1, E2, and E3, and we have specific prosthetic groups associated with each subunit. So we're going to begin with subunit 1. In subunit 1, we are going to be dealing with TPP. So TPP is a very large molecule. Over here, we have the depiction of TPP, thiamine pyrophosphate. So TPP, being a large molecule, what are we interested in? We're interested over here at this negative charge. This negative charge is on a carbon, so we get a stable carboanion. This stable carboanion has the ability to act as a nucleophile and nucleophilically attack the carbon on alpha-ketoglutarate. So this right over here is the drawing for alpha-ketoglutarate. So we're going to nucleophilically attack this carbon. It's going to nucleophilically attack this carbon over here. Okay, the stable carboanion nucleophilically attacks the carbon on alpha-ketoglutarate. Now, carbon can only have 4 bonds. So if TPP is trying to form a bond, that stable carboanion of TPP, if it's trying to form a bond with that carbon we can see already has 4 bonds. It has 1, 2, 3, 4 bonds, 2 bonds over there, right? So it already has 4 bonds. So it must break a bond in order to be able to make a bond with TPP. And it's actually going to break its bond right over here. So we're going to get a release of this COO minus. And what's this COO minus going to make? It's going to make carbon dioxide. So it's going to make CO2. Because remember, the structure of COO minus is simply going to be like this. And this is carbon dioxide. All right? So we get this production of carbon dioxide, and then TPP is able to form a bond with alpha-ketoglutarate that has now lost its carbon and 2 oxygens. So we're left off with this for alpha-ketoglutarate, and it binds with TPP. So we can actually see that we have a different chemistry over here. So over here we can see that we actually had a double bound oxygen, so we had a carbonyl, but we no longer have this carbonyl over here. Instead we have a hydroxyl group. Okay, that's important to make note of. Another thing that is important to make note of is that we no longer have 5 carbons. We have 1, 2, 3, 4 carbons. So we've lost one of our carbons, and this is a very important step because remember, at the beginning of TCA, when we add in acetyl CoA to oxaloacetate, we get a 6-carbon molecule. And we have to get rid of the 2 carbons that we get from acetyl CoA because remember, we want to regenerate everything in the end. So the 2 carbons that we get from acetyl CoA, we want to get rid of them. And this is where we get rid of them. So now we're only left with 4 carbons. We just released one as CO2. Now the next thing to look at is lipolysine. So lipolysine, it's made up of this lipoic acid, which is bound to lysine. So this is just the structure of lipoic acid, and then it's bound to lysine. And I didn't draw the lysine, I simply just wrote it in the center. So lipolysine has a very special role. So remember, lipolysine, it's found in the 2nd subunit. So it's found in E2. And lipolysine is moving around and it sees that TPP has bound with the remaining portion of alpha-ketoglutarate. And it's going to be like, okay, I'm going to pick it up. So it's going to pick it up. But as it does so, we can see over here that we have this sulfur-sulfur bond right over here. So lipolysine is currently in its oxidized state. But when it picks up the alpha-ketoglutarate, the decarboxylated alpha-ketoglutarate, when it picks it up, lipolysine becomes reduced, okay? Because we can see it gained that H, it gains the decarboxylated alpha-ketoglutarate, so it becomes reduced. So this sets up the stage for the next reaction that's about to happen. The next step that happens is we have coenzyme A come in. So coenzyme A is a very large molecule. And I have only drawn out the part that we are interested in. And we are interested in this part right over here. So coenzyme A has this group right over here, this sulfur. So this sulfur is very important because it can act as a nucleophile. It's a good nucleophile. And it's going to nucleophilically attack the carbon right over here. When it attacks that carbon, remember, carbon can only have 4 bonds. So it must break a bond, and it's going to break the bond right over here. When carbon breaks that bond with sulfur on lipolysine, it'll be able to form a bond with this sulfur of coenzyme A. So as a result, we're going to see that coenzyme A has now bound to the decarboxylated alpha-ketoglutarate, and we get the resulting molecule, which is known as Succinyl CoA. Okay, so we got Succinyl CoA. But we need to make note of one thing. When we pass down decarboxylated alpha-ketoglutarate to lipolysine, lipolysine now exists in its reduced state, and we have to oxidize it again so that lipolysine can be ready to pick up another decarboxylated alpha-ketoglutarate. So we need an electron carrier, and we have FAD. So FAD is going to pick up these electrons, and it's going to pass it on to NAD+. Now we may be wondering, Why do we have 2 electron carriers? Why can't we just have one and one does the job? Well, we want NAD plus because NAD plus is able to generate more energy, right? Because NAD plus can generate 2.5 ATP, whereas FAD can only generate 1.5 ATP. So ideally, we want NAD plus. But the problem is that NAD plus can only accept 2 electrons at a time. But FAD has the ability to pick up 1 or 2 electrons at a time. And over here, lipolysine is only passing on 1 electron at a time. So that's why we need FAD. So FAD is going to pick up 1 electron at a time, and it's going to get reduced to FADH2. Then it's going to pass on its electrons to NAD+, and then we're going to get NADH and H+. Okay? So that is the final product of this mechanism, where we get NADH plus H+. Now, if we take a look back, we can actually see that everything in the process was regenerated. So when we had TPP and it bound to the decarboxylated alpha-ketoglutarate, it was regenerated when lipolysine picked up the decarboxylated alpha-ketoglutarate. And in order to regenerate lipolysine, we simply oxidized lipolysine over here and passed on the electrons to FAD. And then we needed to regenerate FAD, so we FADH2, so we passed on the electrons to NAD+, to get NADH and H+. So now, things to just go over quickly is that we do have an oxidation reaction and we do have a decarboxylation. So over here, we can note of our decarboxylation. Here we're actually undergoing a reduction because we're converting the oxidized lipolysine into its reduced state. And then we have an oxidation happening over here. Then we have an oxidation happening over here, and we have a reduction happening over here. So overall, this is an oxidative decarboxylation mechanism in which we are utilizing TPP, coenzyme A, FADH2, and lipolysine to go from alpha-ketoglutarate to succinyl CoA. And that's essentially the mechanism of alpha-ketoglutarate dehydrogenase complex, which consists of 3 subunits, and each subunit has a specific prosthetic group, and 3 prosthetic groups and subunits work together to pass things on to one another efficiently to go from alpha-ketoglutarate to succinyl CoA.